Please.
Hi, thank you so much to be here. I'm Norma Perez, and today I'm going to talk about regulation of calcium homeostasis in the Trans-Golgi network of HeLa cells. I did this work in collaboration with Dr. Isla Kuma from Heidelberg University, and this work was supported by CONACYT. So first, I want to give you a short introduction about why it's so important to study calcium ion. So in our body, there is around 1.5 kilograms of calcium in our bones, and approximately 10% manages to escape from the skeleton and circulates in our body to regulate many physiological processes. Our body contains like 60% of water, so mainly calcium ions are surrounded by water molecules. They interact partially with the oxygen of these molecules. So then the question is, how does calcium enter the cell? In the plasma membrane, there are some proteins. These proteins contain oxygen atoms. So basically, it's through the interaction of oxygen atoms that calcium can penetrate the cell. So outside of the cell, the concentration is high, around 2 millimolar, but inside of the cell, the concentration is 100 nanomolar. So once calcium is inside of the cell, in the cytoplasm, there are a lot of proteins, which also contains oxygen atoms. So through the interaction with oxygen atoms, calcium stabilizes the conformational states of effector proteins, and in this way, many signaling pathways are triggered. But also in our body is full of phosphates. So this calcium then can also interact with the oxygen of these phosphates, and this reaction is cytotoxic. So basically, this reaction means bond. So that's why cells invest a significant amount of energy to keep the concentration of calcium around 100 nanomolar. At cellular level, there are two sources of calcium: the extracellular space and some organelles, such as the endoplasmic or sarcoplasmic or endoplasmic reticulum. As I said, calcium enters the cell through some channels, and here in the cytoplasm, it's full of proteins. Calcium stabilizes their conformational states, and in this way, many signaling pathways are triggered, since exocytosis through fertilization. Here in these organelles that basically keep calcium, there are some proteins that functions as a calcium release channels, such as ryanodine receptor or the IP3 receptor. And also there are some proteins that pump calcium against the concentration gradient to the lumen of the organelle. So this is a diagram, but in order that you have an idea about the distribution of these proteins in the organelles or in some cells, here you are looking at the localization of some calcium release channels, in this case is ryanodine receptors, and here you are looking at the localization of SERCA pumps. And here you are looking at the co-localization of these two proteins. So basically, here, for example, these two proteins are located around the nucleus. So this is mainly, we found in organelles, we found pumps and channels, okay? And also the other thing that we need to know is how is the calcium ion measured. So mainly, calcium can be either free or bound to proteins, right? So adding some calcium sensors, basically we can track the free calcium concentrations concentration in the cytoplasm, but also in the organelles. So once this calcium sensor interact with calcium, some of their properties change, and in this way we can track the activity of calcium. Okay, so this work, or I am interested to study how the level of calcium is kept in the Trans-Golgi network. So I talk about the endoplasmic reticulum, and here the proteins are synthesized and released, and through the secretory pathway. So basically, proteins travel to this Golgi complex and reach the Trans-Golgi network. So my work is here, located in this organelle. We're interested to study how the homeostasis is kept in this organelle. So in this secretory pathway, there is a calcium gradient. So the free calcium concentration in the reticulum is around 400 micromolar, but the free calcium concentration in the TGN, it's around 100 micromolar. So there is a gradient, okay, in the secretory pathway. But also the molecular players that moves calcium in the pathway basically change. So here, for example, in the ER, we have the cerca-POM and IP2 receptors or ryanodine receptors. But here in the TGN, at least this diagram reported that we have the secretory calcium, secretory pathway calcium ATPase, and some as a POM, and some release channels such as the ryanodine receptor. But we study HeLa cells, so we are not sure exactly what are the molecular players or not at this point. So here, you are looking just at tomography of the Golgi complex. And as I said, my work is related, or we are working in this compartment, how calcium is kept there. So up to now, there is a signaling pathway that somehow controls the level of calcium in the Trans-Golgi network. So basically, we have calcium in the cytosol, and calcium enters the cells enters the Trans-Golgi network via the secretory pathway calcium ATPase. And here there is a protein called Ca45. And this protein contains 6 calcium binding sites. So calcium basically interacts with this protein. And then this complex attach another proteins such as a lysosome C or COMT. So in this way, these proteins are sorted. So I'm not sure if you're familiar with the Trans-Golgi network, but mainly the sorting process happens via some receptors. But here, there is a sorting process that is happening via the calcium activity. So once this complex is formed, the phosphorylation of Ca45 by 520 basically detach this phosphorylated Ca45, and in this way, basically the sorting process happens. And all the Ca45 that is unphosphorylated remains in the Trans-Golgi network. So once we basically build the whole picture about the calcium or the calcium toolkit in the Trans-Golgi network, we propose the computational model. So here our model contains 4 reactions. The first one is related with the movement of calcium from the cytoplasm to the Trans-Golgi network. The second one is the interaction of calcium with the Ca45 protein that produces this complex, and then the oligomerization of the complex and the release of this oligomer. And here you are looking the kinetic rate of every one of the reactions and the parameter values. We use COPASI. COPASI means complex pathway simulator to estimate the unknown parameter values. So in order to estimate that parameter values, we use some experimental data reported in the literature. So here you are looking the level of calcium, free calcium in the lumen of the Trans-Golgi network. So this is a control case. Basically, this calcium, the level increased because a pulse of calcium was added to the medium. And then you are looking here the level of calcium under 2 different circumstances. Blue dots shows the level of calcium when the level of Ca45 was decreased. And also you are looking here the level of calcium when the POM was decreased, okay? So then we use a control case to fit the unknown parameter values. And here you are looking the result of our estimation, parameter estimation. So once we had that part, then we can obtain the temporal courses of every one of the chemical species involved in our system. And here you are looking, for example, the level of Ca45 and the formation of the oligomer to the time. And also the level of calcium in the Trans-Golgi network, okay? So once we have this, then we try to reproduce the experiment regarding, this Ca45 knockdown experiment. So in the model, we decrease our concentration around 30%, and we basically follow the level of calcium in the TGN. And we observe that, that the level of calcium increased instead of decrease. And if you don't remember, when we decrease the level of Ca45, basically the level of calcium in the TGN also decreased. But from the model, we are looking that calcium is actually behaving in the other way around. So some part is missing here. So we went into the literature and we tried to identify, I don't know, many elements that are controlling calcium somehow. And we found in this paper that there is a calcium release channel which is sensitive to emetin in HeLa cells. So we don't know the identity of this channel, but probably this channel somehow is working as a functional unit with the Ca45 and the POM. In the way that once the Ca45 is decreased in the experiment, then this calcium release channel is like evident now. And probably that's the reason why we have this behavior. But then the model that we have, we need to do more in this model. And the idea is to add the reaction regarding the calcium release channel, and also to estimate the parameter values and obviously test our proposal. So finally, I want to say thank you to all the persons involved in this work and CONACYT to support this project. And this is a kind of advertisement. I will give a workshop on Sunday regarding Copasic, which is the software that we use to solve this problem. And also, I will present these combined standards, which are standard notations to build computational models regarding or to analyze biological systems. So if you're interested, please be here. And I will give the workshop in Spanish in order that or to reach more audience. So yeah, that's everything that I have.